Welcome back. Today our topic is muscle tissues. I'm going to be talking about types of muscle tissues, and we have been talking about muscle contraction and relaxation, mechanisms of muscle contraction. So, muscle is one of the four primary tissue types of the body. And the body contains three types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. All three muscle tissues have some properties in common. They all exhibit a quality called excitability, as their plasma membranes can change their electrical states from polarized to depolarized and send an electrical wave called an action potential along the entire length of the membrane. While the nervous system can influence the excitability of cardiac and smooth muscle to some degree, skeletal muscle completely depends on signaling from the nervous system to work properly. On the other hand, both cardiac muscle and smooth muscle can respond to other stimuli, such as hormones and local stimuli. So, the body contains three types of muscle tissues. The muscles all begin the actual process of contracting, shortening, when a protein called actin is pulled by a protein called myosin. This occurs in striated muscles, skeletal and cardiac, after specific binding sites on the actin have been exposed in response to the interaction between calcium ions and proteins, troponin and tropomyosin, that shield the actin binding sites. Calcium also is required for the contraction of smooth muscle, although its role is different. Here, calcium activates enzymes, which in turn activate myosin heads. All muscles require adenosine triphosphate, ATP, to continue the process of contracting, and they all relax when the calcium is removed and the actin binding sites are reshielded. A muscle can return to its original length when relaxed due to a quality of muscle tissue called elasticity. It can recoil back to its original length due to elastic fibers. Muscle tissue also has the quality of extensibility. It can strengthen or extend. Contractility allows muscle tissue to pull on its attachment point and shorten with force. Skeletal muscle fibers. Skeletal muscle fibers are multinucleated structures that compose the skeletal muscle. Some other terminology associated with muscle fibers is rooted in the Greek sarko, which means flesh. The plasma membrane of muscle fibers is called the sarcolemma. The cytoplasm is referred to as sarcoplasm, and the specialized smooth endoplasmic reticulum, which stores, releases, and retrieves calcium ions, is called the sarcoplasmic reticulum, SR. As will soon be described, the functional unit of a skeletal muscle fiber in the sarcomere, a highly organized arrangement of the contractile myofilaments, actin, thin filament, and myosin, thick filament, along with other support proteins. Muscle fiber. A skeletal muscle fiber is surrounded by a plasma membrane called the sarcolemma, as we said, which contains sarcoplasm, the cytoplasm of muscle cells. A muscle fiber is composed of many fibrils, which give the cell its striated appearance. The sarcomere, the striated appearance of skeletal muscle fibers, is due to the arrangement of the myofilaments of actin and myosin in sequential order from one end of the muscle fiber to the other. Each packet of these microfilaments and their regulatory proteins, troponin and tropomyosin, along with other proteins, is called a sarcomere. The sarcomere is the functional unit of the muscle fiber. The sarcomere itself is bounded within the myofibril that runs the entire length of the muscle fiber and attached to the sarcolemma at its end. As myofibrils contract, the entire muscle contracts as well. The sarcomere is the region from one Z line to the next Z line, is the functional unit of a skeletal muscle fiber. Muscle fiber contraction and relaxation, the sequence of events that result in the contraction of an individual muscle fiber, begins with a signal the neurotransmitter acetylcholine from the motor neuron innervating that fiber. The local membrane of the fiber will depolarize and positively charged sodium ions enter, triggering an action potential that spreads to the rest of the membrane will depolarize, including the T-tubulus. This triggers the release of calcium ions from storage in the sarcoplasmic reticulum. The calcium then initiates contraction, which is sustained by ATP. As long as calcium ions remain in the sarcoplasm to bind to troponin, which keeps the actin binding site unshielded, and as long as ATP is available to drive the cross-bridge cycling and the pulling on of actin strands by myosin, the muscle fiber will continue to shorten to an anatomical limit. Contraction of a muscle fiber. A cross-bridge forms between actin and the myosin, hence triggering contraction. As long as calcium ions remain in the sarcoplasm to bind to troponin, and as long as ATP is available, the muscle fiber will continue to shorten. Muscle contraction usually stops when signaling from the motor neuron ends, which repolarizes the sarcolemma and T-tubulus and closes the voltage-gated calcium channels in the sarcoplasmic reticulum. When signaled by a motor neuron, a skeletal muscle fiber contracts as the thin filaments are pulled and then slide past the thick filaments within the fiber's sarcomeres. This process is known as the sliding filaments model of muscle contraction. The sliding can only occur when myosin binding sites on the actin filaments are exposed by a series of steps that begins with calcium entry into the sarcoplasm. When a sarcomere contracts, the Z-lines move closer together, and the I-band becomes smaller. The A-band stays the same wide. At full contraction, the thin and thick filaments overlap completely. Tropomyosin is a protein that winds around the chains of the actin filaments and covers the myosin binding sites to prevent actin from binding to myosin. Tropomyosin binds to troponin to form a troponin-tropomyosin complex. The troponin-tropomyosin complex prevents the myosin heads from binding to the active sites on the actin microfilaments. Troponin also has a binding site for calcium ions. To initiate muscle contraction, tropomyosin has to expose the myosin binding site on an actin filament to allow cross-bridge formation between the actin and myosin microfilaments. The first step in the process of contraction is for calcium to bind to troponin so that tropomyosin can slide away from the binding sites on the actin strands. This allows the myosin heads to bind to these exposed binding sites and form cross bridges, myosin with actin. The thin filaments are then pulled by the myosin heads to the slide past the thick filaments toward the center of the sarcomere. But each head can only pull a very short distance before it has reached its limit and must be recocked before it can pull again, and step that requires ATP.